In this video, we'll take a look, an overview of the nitrogen cycle. Nitrogen is present in huge quantities in the atmosphere in the form of molecular nitrogen, N2, N triple bonded to another N, which is a gas. It comprises about 80% of the atmosphere. Despite its abundance, the only creatures that can obtain N2 and turn it into a biologically available form are those prokaryotes of the nitrogen-fixing bacteria class. Those are prokaryotes that possess nitrogenase enzymes. That is the only enzyme that can turn nitrogen into something useful. Now, there's already lots and lots of nitrogen in the biosphere, contained within living organisms. And that nitrogen is recycled also in huge, huge quantities by such creatures as the amonifying bacteria. So you'll notice that both taking inorganic, unavailable nitrogen and making it biologically available, and recycling organic molecules lead to the production of ammonia or ammonium. And ammonia and ammonium are essentially always present in the same places at the same time, and the amount of ammonia versus ammonium is really a function of the pH in the environment. Okay, so both nitrogen-fixing bacteria and ammonifying bacteria are going to place ammonia into the soil. Now, nitrogen-fixing bacteria do it because they need that nitrogen to carry out the manufacture of their proteins. They don't have a source of ammonia from outside. They have to generate the ammonia, which they then turn into amino acids, nucleic acids, etc. This is an exceedingly expensive reaction, and so it is not a reaction that you could say is a harvesting of energy. It's an investment of energy to turn N2 into ammonia. In contrast, ammonifying bacteria, by and large, are using their nitrogen-containing nutrients both as building blocks. They can grab amino acids and turn them into new proteins, or they can grab amino acids and burn them as fuel. When they burn them as fuel, that's when they release ammonia into the atmosphere. Let's take a look at how this occurs in both of these situations. So the nitrogen fixation process requires nitrogenase enzyme. Nitrogenase enzyme is very sensitive to oxygen. If oxygen binds this enzyme, it denatures. It's broken. It's permanently damaged. And so it's essential that oxygen be kept away from the enzyme. On the other hand, it's exceedingly expensive to carry out this reaction. Many, many ATP are required. And so most of the creatures who carry out this process require oxygen to obtain enough ATP from their fuel source, from their sugars or whatever they eat, in order to possess enough ATP to carry out nitrogen fixation. Some free-living bacteria burn a tremendous amount of fuel, much more fuel than they would ordinarily, and carry out much more aerobic respiration than they actually need, and then just essentially waste this fuel on purpose because their object is to get rid of oxygen by using tremendous quantities of oxygen in their electron transport chain, and that reduces the oxygen around so it doesn't destroy the nitrogenase. A very elegant solution, however, is found in plants. In plants that house nitrogen-fixing bacteria, notably the legumes, nodules are formed, so low oxygen concentrations are present, and the plant provides the bacteria with the protein leg hemoglobin, which grabs all of the oxygen that the cytochrome oxidase isn't using, keeps it away from the nitrogenase, and then as the oxygen around it is used up, the leg hemoglobin supplies the cytochrome oxidase with the oxygen it needs. Remember that this is a matter of having an intermediate affinity for oxygen, greater than that of the nitrogenase, less than that of the cytochrome oxidase of the bacteria. So nitrogen-fixing bacteria create ammonia from nitrogen gas. In the ammonifiers, you're also going to see, and in your digestive system, you're also going to see that ammonia and ammonium are made. Now, when you eat a high-protein meal, some of that protein, some of the amino acids that you absorb are going to be used as building blocks, but some of it will also be used as fuel. How do you use an amino acid as fuel? Well, the amino group is taken off, and that leaves us with an ammonia or ammonium, and alpha-ketoglutarate is produced, at least when glutamate is the starting amino acid. You notice that alpha-ketoglutarate is a component of the Krebs cycle, the citric acid cycle. So the more you can plug into the citric acid cycle, the more NADH is produced, the more FADH is produced, FADH2, and so the more ATP is going to be made by the electron transport chain. So you don't have to put the amino acid through glycolysis, obviously you plug it right into the citric acid cycle. So there's glutamate being used as a fuel. Glutamate, of course, could also be put into another another protein, so it could be used as a building block. One of the ways that ammonia is toxic is it forces this reaction to run backwards. When there's lots and lots of ammonia, alpha-ketoglutarate and ammonia become glutamate. So lots of this amino acid is produced in order to get rid of this ammonia, and the cell begins to starve. It doesn't have the alpha-ketoglutarate to run the Krebs cycle. Even if you produce more, if it's being turned into alpha-ketoglutarate and alpha-ketoglutarate is being turned into glutamate, then there's not enough ATP being made, and that can cause brain damage in neurons that rely on the citric acid cycle to provide their ATP. Okay, so ammonia then is provided by both of these bacteria into the soil. Now, ammonia or ammonium is an energy-rich food for the nitrifying bacteria. Nitrifying bacteria consist of two classes. There's the ammonia oxidizing, and then there's the nitrite oxidizing bacteria. And in combination, the end product of nitrification is nitrate. Okay, so let's take a look at the ammonia. I'm going to get rid of this. At the ammonia bacteria's use of, the ammodifying bacteria's use of ammonia. There's the ammonia, and it's being plugged right into the electron transport chain. The electrons and the protons from the ammonia are being donated to the electron transport chain, and a molecule that's going to end up as nitrite is provided as its end product of this reaction. So what happens to the electrons and the protons? Well, the electrons run through the electron transport chain, providing a proton gradient that is used to generate ATP using ATP synthase. Notice that cytochrome oxidase is part of this electron transport chain, and that therefore oxygen is required in this process. Okay, then that nitrate is then further oxidized into, in a different bacteria, nitrate. And then that nitrate is found in the soil. What becomes of the nitrate? Well, plants love to take up nitrate. That is their most common nitrogen-containing nutrient. Plants take it up and then turn it back into ammonia, and then turn it back into amino groups and nucleic acid, amino groups and nucleic acids as well. And so that's being used as a building block. Plants can also use ammonia as a building block. But sometimes, so most of the ammonia that gets dropped into the soil is converted by these nitrifying bacteria, using it as ammonia, a building block, but also as their primary source of calories, so that most of the ammonia gets turned into nitrate in most environments. Now, the denitrifying bacteria, they can use nitrate as a building block. They can use ammonium as a building block. But they can also use nitrate as a substitute for oxygen when the soil becomes anaerobic. So they prefer to do aerobic respiration because in aerobic respiration they can use cytochrome oxidase, use oxygen as the final electron acceptor, and therefore have 3 separate proton pumps within their electron transport chain. They're going to get a lot of energy out of the sugar fuels that they consume because they get lots of energy out of their NADH, just like we do. But when oxygen isn't present, they have an alternative pathway, an anaerobic respiration system they can use. They can use nitrate as the replacement final electron acceptors. The electron ends up here. And the 2 pumps are still available earlier in the electron transport chain from their fuel. Now they do lose efficiency when they do this because the nitrate reductase that accepts the nitrate, that reduces nitrate, provides the electrons, or removes the electrons from the chain so that it can continue, puts them onto the nitrate. That is not a proton pump, and so you don't get as much proton motive force, so you don't get as much ATP per unit NADH, per unit fuel. So this is an anaerobic respiration, not as efficient as aerobic respiration in these creatures, but much, much more efficient, provides much, much more ATP than would fermentation. There's your nitrogen cycle. I guess we're back here in this because we've eaten all of the plants that have turned all of these ionic forms of nitrogen nutrients back into amino acids.